Thank you for joining us today at our drug discovery and development event. I just want to say a brief word, or brief words, to introduce Dr. Chahwan before he begins his quite fascinating presentation. So after completing his undergraduate degree in Physiology and Biophysics at the University of Toronto, he spent 5 years at the Scripps Institute examining mechanisms of cell cycle and checkpoint control, DNA repair pathways, and genomic stability in both healthy and cancer cells. He then returned to the Department of Molecular Genetics at the University of Toronto, where he earned his doctorate studying additional mechanisms responsible for controlling genomic stability, aneuploidy, and chromosome transmission. And whilst in academia, he was the recipient of multiple governmental grants and has authored over 20 peer-reviewed publications. So approximately 5 years ago, Dr. Chahwan co-founded SyntheX. And as its chief scientific officer, he has spearheaded the company's efforts to examine intracellular targets. And these are targets that have been selected based on oncogene addiction and synthetic lethality related properties. Through the development of SyntheX's Torpedo and Tornado platforms, which he will be mentioning in his presentation. So these drug discovery and developmental platforms have uncovered peptide and macrocycle candidates that act respectively to disrupt protein-to-protein interactions or to degrade neo-substrate targets. Using Agilent's label-free Excelligence real-time cell analysis instrumentation to drive SyntheX's core screening efforts, these platforms have fed the company's therapeutic candidate pipeline, which is predominantly focused on targets that have been previously deemed to be undruggable. So Dr. Chahwan's talk today, which is entitled Using an Unbiased Drug Discovery Platform to Uncover a Novel Immunogenic Cell Death, will provide an overview of SyntheX's lead candidate, STX100, from discovery through preclinical efforts and to the cusp of clinical development. And with that, I will pass it over to Dr. Chahwan.
Hello, everyone. My name is Charly Chahwan. I'm the CSO and co-founder of SyntheX, a biotech startup in San Francisco. First, I'd like to start with thanking the organizers for the invitation, and I'd like to thank you all for tuning in today. At SyntheX, we are building a collection of synthetic biology technologies to enable us to produce and functionally select potent compounds that are capable of targets that are thought to be or canonically conventionally assumed to be undruggable targets. What I mean by undruggable are basically targets that have so far eluded drug discovery approaches. These types of undruggable targets include protein-protein interactions, protein-DNA interactions, protein-RNA interactions like transcription factors or splicing factors, as well as low specificity enzymes like phosphatases, nucleases, esterases in general. And the conventional wisdom in the field is that currently these types of targets are undruggable, are out of reach because the available libraries of chemical compounds that we have at our disposal are not complex enough to be able to contain compounds that can efficiently engage these targets, these difficult and challenging targets. And the other thing is that even if we were to overcome that limitation of lack of complexity, if we were to achieve somehow this level of complexity, the labor-intensive nature of the screening approaches required for screening for these types of activities is quite prohibitive. So what we've done at SyntheX is basically we've decided to rely on the power of synthetic biology to specifically address these two challenging tasks. And what we've done to address these specific two things, first of all, to address the question of labor-intensive screening approaches, we've built a set of synthetic circuits that are proprietary to us. And we utilize these circuits to allow us to screen for complex biological functions in a facile and cost-effective way. And on top of the logic circuits that were built in our platform, we actually include the ability to make a very complex library of over 10 billion different compounds that are actually being made inside the cells off of the ribosome. So we combine these two innovations in one of our platforms that we call Torpedo in order to be able to identify factors or compounds that can disrupt intercellular protein-protein interactions. On the other hand, with the Tornado platform, we rely on these same two innovations, but in the case of Tornado, instead, the question is not can we disrupt two protein-proteins from interacting with each other, but rather can we bridge a ubiquitin E3 ligase to a particular target of our choice, of our interest, in such a way that the target now becomes a neo-substrate for the E3 ubiquitin ligase, and the ligase ubiquitinates it and subsequently targets it to the proteasome for it to be degraded by the ubiquitin proteasome system. And the programs in our company that rely on Torpedo right now are two programs, RAD51 and oncogenic KRAS programs. RAD51 I'll talk about in much more details in a second. KRAS would be beyond the scope of this talk. And we do have another couple of programs that rely on the Tornado technology. So moving on to RAD51, in this target, basically I've been interested in for more than 15 years now. The interest stemmed originally from these foundational publications that came out from the Halliday and Alan Ashworth labs in the UK in 2005. In these publications, the two labs reported that they've discovered the fact that breast cancer cells that are mutated in the BRCA1 or BRCA2 genes are particularly sensitive to inhibition of an enzyme that's known as poly-ADP ribose polymerase, or shortened for PARP. And what they found is you could either genetically ablate by knockout the PARP gene and then cause death of these BRCA mutated cancer cells, or if you use a small molecule inhibitor of the PARP enzyme, you specifically cause death of the BRCA mutated cancer cells, but not of healthy BRCA wild type or BRCA heterozygous cancer cells, intact cells. And fast forward for less than 10 years, a bunch of pharma companies got these types of small molecule compounds that target PARP FDA approved, and we now have 4 of them in the clinic that are FDA approved for multiple indications, where the cancers have a BRCA1 mutation or BRCA2 mutation, or mutations in any of the components that are cofactors of the BRCA pathway genes. However, as is always the case with precision medicine, this has been one of the recent triumphs of precision medicine, but there's always caveats. And the caveat here in this particular case, obviously, is that resistances do arise in different ways. We knew about several ways how resistance could arise in the pathway because of mouse models of breast cancer and BRCA mutations. There are many different approaches, different ways how cancer cells in the mice or in tissue culture end up becoming resistant to PARP inhibitors, cancer cells that carry BRCA mutations. But in the clinic, on top of all these different paths that resistance could arise, in the clinic, there are one other key factor that seems to be quite common and one of the easier ones to attain, and that is the upregulation of a protein called RAD51. So it is even before administration of PARP inhibitors, people had noticed, multiple scientists have noticed that in BRCA mutated cancers, there is high upregulation of the RAD51 gene, and that gives fitness to the BRCA mutated cells. And more recently, after PARP inhibitors got into the clinic, studies have uncovered from among some of the PARP resistant patients, a group of patients that downregulate a protein called the MI1, which is an ubiquitin ligase that causes degradation of the RAD51 protein. So a cancer cell could either upregulate the transcription of RAD51 or cause the stabilization of the RAD51 protein by inhibiting its degrading factor and become resistant to PARP inhibitors. And some cancer cells might rely on both approaches, or even more further stabilizations by further post-translational modifications. And that phenomenon of upregulation or hyperstabilization of RAD51 is actually not unique to PARP inhibitors or to BRCA mutated cancers or to cancers that have been subjected to a selective pressure like with a chemo drug. But rather, it's a very common phenomenon that happens quite often in cancers. It's been reported in metastatic breast cancer, in prostate cancer, in pancreatic cancers, in a lot of hematological cancers. It's a very common occurrence, just like it's almost analogous to the occurrence of having telomerase being re-induced in the cancers relative to adult tissue. And the other thing about RAD51 that is very exciting to us is that it is very lowly expressed in adult healthy tissue. So you can see here, this is a northern blot showing RAD51 in one of its cofactors, the mRNA levels of them. And you can see here that in most adult tissues, except for the male germline testes, there is no detectable levels of the RAD51 protein. And obviously in the male germline, it's expected to see RAD51. That RAD51 is a recombination protein, homologous recombination protein. And in the male germline, there's always recombination happening during spermatogenesis. So one would expect to see high levels of RAD51 there. But otherwise, it is a very lowly expressed protein in healthy tissue. However, in cancer tissues, in this case down below here, I'm pointing out to multiple different cholangiocarcinoma liver cancer cell lines that have very high levels of RAD51. Yet the normal tissues, in comparison, have no detectable RAD51 at this level. And that's not restricted to the mRNA. We see that in the lab also with many different cancer cell lines. All of them have detectable levels of the RAD51 protein. This is a Western blot with showing the RAD51 protein here. However, primary cells that are not transformed, not immortalized, have very low levels of RAD51, barely detectable at all. However, whenever these primary cells actually do get immortalized and become closer to becoming cancer, they start showing higher levels of RAD51. And this feature of RAD51 being so uniquely expressed or so strongly expressed in cancer cells relative to healthy cells has been exploited in gene therapy type approaches, where people have done two different things. One, to link a visualization reporter to a RAD51 promoter in order to mark the cancer cells and visualize them in an organism, like in a mouse. And the other approach was to drive a toxin using the RAD51 promoter and delivering that toxin into systemically administering it to a mouse, for example, and seeing that only the tumors shrink and there's no adverse effects of killing of general cells, healthy adult cells in the mouse. And like you can see here in these mouse images, the mice that had tumors implanted in them light up whenever they get given this transgene, luciferase, RAD51-driven luciferase transgene. The mice that did not have tumors implanted in them do not light up at all. There is no particular tissue or compartment in a healthy animal that would have active expression of RAD51 to the level that would be visible by imaging systems. So given this uniqueness of RAD51 expression and abundance to cancer cells, we thought, besides the fact that it's a way how cancer cells gain resistance against PARP inhibitors, it could be a good target that allows us to get selectivity for the cancer cells over healthy cells when we are treating a tumor. And in addition to that aspect of RAD51 being enriched in expression-wise in tumors, there's one other thing about tumors that we tend to forget, and that is tumors, as they're dividing very fast in an uncontrolled and unQC'd way, they end up accumulating a lot of genomic instability in their DNA. And that genomic instability leads to fragments of DNA being extruded out of the nucleus into the cytoplasm. And under normal condition, DNA that is in the cytoplasm activates innate immune responses because we are designed to, our cells are designed to think that any type of foreign DNA that's residing in the cytoplasm, and any type of DNA residing in the cytoplasm is going to be of foreign origin, like an adenovirus or some other DNA-based virus that came to attack our cells, and we should mount an immune response against it. So in cancer cells, because there's a lot of DNA that's not just in the nucleus, but also in the cytoplasm, usually you'd think that these cancer cells would be mounting constant inflammatory type responses because of that extra DNA activating the innate immune response in them. However, that doesn't happen because of RAD51. RAD51 has the ability to coat this single-strand DNA and forms a filament around it and prevents it from engaging the innate immune response DNA sensing machinery. So just, just as a summary about what we know about the RAD51 protein at the biochemical level, it's an ATPase. It hydrolyzes ATP, it binds it and hydrolyzes it and controls its activity that way. It forms a nucleofilament around single-strand DNA, just like actin forms a filament in our cytoskeleton or microtubule, same thing. It's very similar to these types of motor proteins. And it is the ATP-bound form of RAD51 that is the active polymer-forming form that binds to single-strand DNA. And this form specifically binds to single-strand DNA when it's in the calcium binding form, so the calcium-bound form of the triphosphate state of the ATPase. RAD51 does get loaded on DNA via the BRCA2 protein, and that's why overexpressing it. BRCA2 basically catalyzes the loading of RAD51 onto DNA. And that's why whenever there's a BRCA mutation, you can bypass that mutation by having high levels of RAD51 compensating for the weak catalysis. RAD51 can also bind double-strand DNA. It actually binds single and double-strand DNA, basically at the junctions particularly. Gets loaded, as I said, by BRCA2. It has intrinsic GTPase activity, and it controls itself just like many other types of nucleotide hydrolyzing proteins like GTPases, like KRAS and others. They auto-regulate themselves by hydrolyzing the ATP and converting it to ADP. And that ADP-bound form in this particular case for RAD51 is the inactive form that is incapable of productively engaging single-strand DNA. We are not the only people who actually thought about RAD51 as being a good target. There has been multiple attempts by multiple different labs, academic labs and industrial labs, trying to find compounds that engage RAD51. A lot of attempts have gone after its single-strand DNA binding activity. Others have went after its double-strand DNA binding activity, and some attempts have gone after its ability to form polymers, basically to break its own polymerization potential, the interactions between two RAD51 protomers. From all these attempts, there's only one compound that ended up going to the clinic, and that's Cytera Therapeutics compound. Cytera is in Boston. They just went into the clinic just a few months ago, and their compound basically works in a bit of an unclear way, but what is known is that it prevents RAD51 from migrating to sites of DNA damage where it needs to act and promote repair. And yeah, that's I've just lighted up the clinical trial that Cytera is currently undertaking. So given all that precedence and the lack of success with all these different approaches, we decided to take a little bit of a different approach at SyntheX, especially relying on the fact that we can have the ability to break protein-protein interactions in a very good and efficient way. And one of the things that caught our attention from the gene expression profiling that was done a while back was that one of the common things that happen with BRCA-mutated cancers is the upregulation of RAD51, but the upregulation of RAD51 does not happen in a void, does not happen on its own. It tends to get upregulated along with several other co-effectors or effector proteins that help it work. And while it has many other effectors, several co-players working with it, particularly RAD54 and RAD51AP1 are the two genes that get co-upregulated whenever RAD51 gets upregulated in BRCA-mutated cancers. So we thought maybe there's something particular about the interaction of RAD51 with these particular proteins that is especially important for cancers in general, and particularly cancers that are BRCA-mutated or that have been exposed to PARP inhibitors. So our approach basically boils down to, can we break the RAD51-AP1 interaction? And if we can, what sort of phenotype can we get in inhibition of the RAT51 activity can we get out of that. So we relied on our torpedo platform to discover protein-protein interactions of the RAT51, RAT51 AP1 protein-protein interaction. And those types of screens yielded several hits. We underwent a prolonged effort to perform SAR and understand the mechanism of how these compounds are acting on RAT51 and how can we improve their pharmacokinetic properties, and ended up with a very stable derivative of one of our hits that we biochemically stabilized and took forward to biochemical studies and to tissue culture studies and in vivo studies. And I'm going to be presenting some of this work right now. So starting with just binding, one of these hits that we found is an L-amino acid peptide, came out straight out of the torpedo platform. It binds in single-digit nanomolar affinity to RAT51, very good binder. We've managed the final compound that was the result of our SAR campaign is a hyperstabilized D-amino acid peptidomimetic that's very stable, yet retains the same level of affinity to RAT51 as the initial starting tool compound that came out of the screen. And one of the nice things that we've managed to find is a single substitution version of the peptide that completely loses its ability to bind RAT51. And this version of the compound has become quite crucial for our studies subsequently, in order for us to know that the phenotypes and the effects we're seeing are on target. Whenever we use the compound that cannot bind RAT51, we don't usually see any of the effects we see with this compound. So to start with, after we got the stable compound, we wanted to see whether it's being able to enter cells. Again, peptides have a bit of a bad reputation about not being able to get into cells very easily. But we were lucky and fortunate in this particular set of peptides that they were pretty cell permeable on their own. So in this panel here, I'm showing a FITC labeled version of the active peptide, as well as the inactive peptide. STX100 M is the inactive one. Both peptides enter cells, and we can see them green inside the cells. The active peptide that binds RAT51 enriches in the nucleus, and that makes sense because that's where RAT51 is. The inactive peptide enters the cells, but it remains pancellular, doesn't actually go into the nucleus, and that also makes sense. If there isn't much RAT51 in the cytoplasm, most of it is in the nucleus, we wouldn't expect to see the peptide going into the nucleus. And then as we started to look at the cells and characterize what happens to the cells when they get exposed to our compounds, one thing that was very peculiar jumped at us very early on, and that is that quite early after treatment of cancer cells with our compound, we do see a very strange hypercondensation of chromatin, and the nuclei become much more hypercondensed, as well as the general integrity of the cytoskeleton of the cells get compromised. There's a catastrophic destruction or depolymerization of most of the microtubule networks, and we end up with cells that look pretty sad, and their nuclei are hypercondensed. That's specific to the active peptide. When we treat the cells with the inactive version of the peptide, we don't see any of these phenomena. And again, I want to point out that this phenomenon happens within minutes. These pictures were taken 30 minutes after exposure to the compound, but we do already see features and phenomena like these in the cells as quickly as 2 or 3 minutes after exposure to the peptide. And because it was such a fast phenomenon, we decided to rely on a cell viability measurement approach that can take very high resolution time-wise detectable time points for viability in our systems. We initially tried a few of the imaging systems. While they're great for seeing what's happening to the cells, they're a bit too slow for a phenomenon that's happening in the level of seconds and minutes. On the other hand, Excelligence from Asaya was a perfect tool for a perfect instrument for us to be able to test and understand further what's happening when our compound is administered to cells. What you can see here, basically, I'm showing you SSP25. These are liver cancer cells that got treated with the active peptide in red. And there's a very quick precipitous loss of cell index and impedance, which is what the Excelligence is measuring. However, treatment with the inactive peptide does not cause any sort of decrease in impedance towards the cells, and mock treatment with PBS doesn't cause any of that either. So one question here that always arises with impedance measurements is that, oh, what if this is not actual death, but rather the cells just underwent some sort of a morphological change that changed their ability to impede the current in the well, and that's why you're seeing this precipitous impedance. This is not death, this is just reflecting morphological changes. So in order to convince ourselves that this is not actually just morphology, but a point of no return type of commitment to loss of life, we went back to microscopy, and we wanted to know whether treatment of cells that overproduce RAD51, like cancer cells, versus cells that do not overproduce RAD51, like primary cells, how do they respond when we treat them to the peptide, and what do they look like when they get exposed to the peptide? And as you can see with these phase images, the liver cancer cells, within 5 minutes of being exposed to the compound, they get these big bubbles forming. The whole cell coalesces around the nucleus, and then the cellular volume forms these big bubbles that extrude out from one corner usually. In 10 minutes in, the cells are looking very, very sad. And that goes on for basically, and they never recover from it. Fibroblasts, on the other hand, the primary cells, they get exposed to the peptide, in this case, compound, and they don't mind it the least bit. You can look at them 5 minutes, 10 minutes, half an hour, 24 hours later, and they're just fine and happy. So that convinced us that what we're seeing is some sort of a programmed mechanism, not just like, oh, our drug is just like bleach, it's just killing cells willy-nilly without any sort of selectivity first, and it's killing cells not in any particular programmed way. Cells die in different ways. It wasn't the case. Every different cancer cell, we tried more than 30 different cancer types, and every single cancer type that got exposed to our peptide, cancer cell type, ends up dying with the same type of morphological features. The DNA coalesces inside the nucleus and hypercondenses. Most of the cell material condenses around the nucleus, and then there's this strange bubbling that happens in a very short period of time. That's not similar to blebbing or necrotic type blebbing or blebbing that is seen in apoptotic cells, let alone the fact that it happens so much faster than the type of bubbling that happens in apoptotic or necrotically dying cells. And like this is just an typical image of how these bubbles would look. The cell content coalesces, and then the cells extrude these bubbles out, and that's happening within 5-15 minutes after exposure to the peptide. So the first question was, maybe what we're doing is the peptide is somehow compromising the integrity of the membrane of these cells, and that's causing them to somehow lose membrane potential and cause sort of osmotic imbalance that's causing this massive bubbling to happen. So in order to know whether the integrity of the cell membrane has been compromised at all, we decided to rely on the lactate dehydrogenase assay. And lactate dehydrogenase is a cytoplasmic enzyme. It's a big protein, so it's not passively diffusing across the membrane. If we take media or supernatants from outside of the cells and assay them for lactate dehydrogenase activity, we shouldn't see any, because the enzyme is inside the cells. As long as the cells are intact, they're not releasing it to the outside, there shouldn't be any activity. But if we lyse the cells with like Triton, any typical lytic agent, detergent of some sort, then we're lysing the cells open, and the enzyme gets outside into the media. And when we measure lactate dehydrogenase activity in the media, we can see very measurable, easy activity. So the control here is Triton, the lysis Triton X lysis buffer, and the lysis agent. On the other hand, if we treat cells with STX100, there is no detectable LDH being released into the outside of the cells. And what was shocking and impressive to us was that when we looked at these cells 24 hours later, we still couldn't detect any lactate dehydrogenase activity. So they were still retaining integrity of the membrane in such a way that was not causing proteins to leak outside of the cells. But then if we were to treat those cells now with a detergent, Triton, then we end up with release of that lactate dehydrogenase into the media. So the cells had died or had committed to a point of no return 5 minutes after exposure to the compound, yet 24 hours later, they had retained the enzymes inside the cells in a functional active state. So it's unlikely that the pH is massively changing in these cells, or there's huge differences in chemical nature that would cause aggregation or instability of a lot of proteins inside the dying cells, the dead cells. So then we concluded that it's not some sort of general membrane destabilization. Then even though, again, the morphology didn't correlate with types of morphology seen with canonical cell death like apoptosis or necrosis, we still wanted to make sure that the machinery that's involved in these types of cell death is not involved either in this new mechanism of death that we've uncovered. And so we went first for necrostatin, which is a RIP kinase inhibitor that would block processes of necroptosis. Here, what I'm showing in green are cells that were treated with STX. This is the impedance measurement with the excelligence. They get treated with STX100, they quickly die. When they are treated with necrostatin, they don't show much death at these time points. If we treat them with a compound along with necrostatin, STX100 along with necrostatin, no abrogation of the cell death is seen. We don't rescue that killing that's caused by STX100. So clearly, RIP kinase and the RIP kinase pathway is not involved in the STX-mediated cell death. Similarly, when we use a pan-caspase inhibitor, again, combining STX100 with the caspase inhibitor here in blue does not alleviate in any way the killing of the cells of the cancer cells that the STX100 does. So to cut a long story short, after a whole bunch of analyses, we concluded that it's the process of cancer cell deaths that we've uncovered is caspase independent, RIP kinase independent. It's not just general membrane destabilization or lysis. It's not ferroptosis, not autophagy, and not mitophagy. And that left us scratching our heads thinking, what is happening here? This is clearly an on-target mechanism that is specific to cancer cells, yet it's a new phenomenon that hasn't been, to the best of our knowledge, reported in the literature. And so we decided to go back to the biochemistry of RAD51 and try to understand exactly what could be causing this, knowing what we know about RAD51's activity. So first, one of the peculiar things that caught our attention during our SAR campaign is that the compounds that we've discovered in our screen bind very well to RAD51 in the single-digit nanomolar range, but they're completely incapable of binding to the homologs of RAD51, orthologs of RAD51 in many yeasts, including budding yeast, the budding yeast Saccharomyces cerevisiae and Gloverensis lactis. So that was particularly peculiar for us because one thing along the same lines that was noted a few years ago was the fact that RAD51 relies on calcium for its activity, yet the yeast RAD51 homolog does not rely on calcium for its activity. It rather uses magnesium for activity. And RAD51 is capable of using magnesium. However, and that's the divalent cation that actually helps it, stimulates its ATPase activity. It helps it hydrolyze the gamma phosphate to make ADP out of it. And what has been shown is that when magnesium is added along with calcium, calcium actually competes with magnesium and prevents the ATP activity of RAD51. And if you recall at the beginning, what I was talking about RAD51 is that it forms this nucleofilament around DNA that coats single-strand DNA. And that nucleofilament is dependent on the ATP-bound state of RAD51. So in that sense, it's also dependent on being calcium bound as opposed to magnesium bound. Because if RAD51 is magnesium bound, it's going to hydrolyze that ATP and it's going to fall off of the denucleate and fall off of the single-strand DNA. So it is very important for RAD51 to be able to bind to calcium in human cells, the human Rat51 protein, yet not the yeast protein. And our peptides, our compounds that we discovered, also bind only to the human protein, but not to the yeast protein. And what we also know about Rat51, about the activity of our peptide on Rat51, is that it is assisting in ATPase activity in ATP hydrolysis. So the thought was, when we actually treat these cells with our peptide, the peptide goes into the cell, the compound goes into the cell, it engages Rat51, stimulates its ATPase activity, and in spite of the presence of the calcium ion. And because of the stimulation of the ATPase activity, the polymer depolymerizes, falls off, and now Rat51 is in the ADP bound state. It no longer needs calcium, and it releases calcium very quickly. So the big question is that what that we posed was, okay, so if our peptide is causing calcium to fall off of the pool of Rat51 that's present in the cancer cells much more abundantly than in healthy cells, is that spike in calcium what might be causing these cancer cells to die? And that was a thought in our minds, because calcium is particularly tightly controlled in its abundance inside the cell, its concentration inside the cell, where it's free. The concentration of free calcium inside the cell is around 50 to 100 nanomolar resting state in a healthy cell. It is around 2 millimolar outside the cell. So there's over 10,000x gradient across the membrane. There's so much active processes that make sure that calcium is not coming from the outside of the cell to the inside, given how big of a gradient there is there. Magnesium, on the other hand, is much more abundant inside the cell, yet there is not much of a gradient between the inside and the outside of the cell. So it was very peculiar to us as to, like, how could it be that Rat51 inside a human cell is utilizing calcium rather than magnesium, even though there's like 10,000 times more of the magnesium cation than the calcium cation in the cell. There must be something active about this process. And there's a mechanism by which the filament is making sure that it stays in the calcium bound state. And if the filament is abundant in the cancer cells, it is acting as a sponge that is soaking up a lot of calcium within it. What we know about Rat51 abundance in murine embryonic fibroblast is that it is in the micromolar range. In the cancer cells, it's presumed to be even higher than that. So if our peptide is capable of engaging the pool of Rat51 in a fast manner, stimulating ATP hydrolysis and releasing all that calcium all at once, we will get at least a 10x, maybe even a bigger 20x to a 50x sudden surge in calcium concentration inside these cells. And that has been our working hypothesis at the beginning, that the cancer cells are dying because we are overpowering the cancer cells buffering capacity, calcium buffering capacity, with this huge surge of calcium. So in order to test that, we first check to see if whether there is an actual calcium spike that happens in the cells that get exposed to our compound. And gratifyingly enough, that's exactly what we see here. We're using the Fluo 2AM calcium sensing dye. And right at this exact same time that we start seeing the cells having problems, we see a very clear increase in intracellular calcium levels. And it's always very consistent, where we see the calcium seemingly arising from the perinuclear region somewhere around the ER, and it emanates from there and fills up the whole cell within 10 minutes. And so if that calcium spike was causative rather than an effect, one of the many effects of the death, one would expect that if it is the cause, then quenching that calcium and mopping it up out of the cell again would be one way how one could be able to rescue these cells from being killed by a peptide. And that's exactly what we see when we rely on, when we use BAPTA-AM, which is an intracellular calcium chelator. Obviously, chelating intracellular calcium is going to be toxic on its own. We see here the impedance being not as good as mock treatment with PBS. The STX100 kills the cells very quickly. The beautiful thing is treatment of the cells along with the peptide, along with a calcium chelator. We get full suppression of that killing that we see. So we know now that it is a calcium-dependent killing process, and that's the only agent that we found that is capable of suppressing the cancer cell death that's induced by STX100. we've also, I'd like to note here that we've also tried chelating calcium on the outside of the cells with EGTA or cell impermeable calcium chelators. And treatment with those types of factors do not at all prevent the killing by STX100. So we do know that the pool of calcium that is spiking up is not coming from the outside of the cell, it's actually coming from intracellular source inside the cell. And that phenomenon of calcium surge got us very, very excited, because a few years ago now, work from the Cromer and Zitvogel lab in France to try to understand how mechanisms of immunogenic cell death arise and what are the compounds that can induce an immunogenic inflammatory state around in the tumor cells and cause engagement of the immune system. In those types of studies that they've done and the screens that they've performed, the strongest agent that they've uncovered that can do this is a set of compounds that are known as cardiac aminoglycosides. And these compounds block sodium-potassium pumps in the cell membrane of cells, cancer cells in that case. And by blocking those pumps, it causes imbalance in the ionic potential across the membrane, and that leads to a very sudden influx of extracellular calcium into the cell. And the surge of calcium inside the cell causes a strong endoplasmic reticulum type stress, UPR type response, ER stress response. And this ER stress response leads to the presentation of immunogenic molecular patterns on the surface of cancer cells. And those molecular patterns are recognized by the immune system, by macrophages and the dendritic cells and professional antigen-presenting cells, and helps them in presenting the neoantigens and antigens from the tumors to the immune system. So we wanted to know whether, when we're killing our cancer cells so quickly with our STX100 compound, are we in the process not just killing them, but also making them present immunogenic markers on their surface that could help the immune system recognize them even faster and quicker and to clear them. And in order to test that, we first checked for one of these hallmarks of immunogenic cell death, a presentation of the calreticulin protein on the surface of the cells. Calreticulin is a protein that's usually found in the endoplasmic reticulum. It's not on the outside of the cells. However, when cells are dying in an immunogenic fashion, they do present calreticulin on their surface. And calreticulin is a marker on the surface of cells that is known as an eat me marker. It basically stimulates macrophages and other phagocytic cells like dendritic cells to phagocytize any cell that is presenting calreticulin on its surface. And so the phenomenon that we see with STX100 is not limited to calreticulin. We see also relocalization of HMGB1, which is a nuclear protein that binds to DNA from a nucleus where it forms usually foci in healthy cells to the pericytoplasmic region all over the cell. So you can see it here in mock-treated cells. The foci of HMGB1 are very distinct and clear, and in STX-treated cells, there is no more foci in the nucleus. It's all spread out all over the cell. And this is one of the hallmarks of immunogenic cell death as well. And this phenomenon happens very, very quickly, at exactly the same time frame that the cell death is happening, the irreversible cell death that we see with the excelligence impedance, sharp drop-off in the impedance. Within 5 minutes, the relocalization has already happened. By 10 minutes, it's reached its maximum. And so now we know that STX100 is killing cancer cells, specifically in healthy cells, by exploiting that cancer-specific massive pool of RAT51 that's acting as a calcium sponge. It engages that pool, releases all that calcium, causes a calcium surge. The calcium surge kills the cells, but also not just kills the cells, causes them to present immunogenic markers on their surface. And those immunogenic markers are known to help engage the immune system in clearing dying cells or compromised cells. And because of all these exciting factors that we see in tissue culture, we decided to now move on and see what do we see in a living animal that gets exposed to STX100 and that has a tumor growing and gets exposed to STX100. And quite gratifyingly, when we moved on to test this in an immune proficient type of cancer model, a syngeneic model, CD26 colon cancer in bulb C mice, It's a very aggressive tumor. These are tumors. These are mice that were treated, just mock treated with PBS. The tumor very quickly gets to volumes, thousands of millimeters cubed of volume within a couple of weeks. The mice that had been implanted with the tumor, and then 9 days later, when the tumor got to around 100 millimeters cubed, they got treated with a single dose of STX100, 400 micrograms of STX100 intratumorally. 50%, 3 out of 6 mice fully resorbed their tumor and got fully cured, and that cure was long-lasting. They never researched and had a tumor regrowth, these tumors, as we followed them. If we were to have used a higher dose of STX100, again, single treatment, but double the dose to 800 micrograms, 5 out of 6, more than 90% of the tumors got fully, completely destroyed, and the mice were fully cured. This type of cancer, again, is extremely harsh, and one of the hallmarks of like the superstar compounds, anti-cancer agents that are in the clinic now are anti-PD-1 and anti-CTLA-4 antibody therapies, Keytruda and Opdivo. In this case, the mouse equivalent of Keytruda doesn't do anything to alleviate the cancers in these CD26 model. However, if we use Keytruda equivalent with the suboptimal concentration of our peptide, we get 90% cures. And obviously, if we use it with the high concentration of the high amount of our compound, we're more or less at the same level. We're pretty much maxed out on how much we can do. And this is just here a presentation of a Kaplan-Meier representation of what we see. And the really exciting thing for us was that we waited for these mice that were cured of their tumors. We had 22 in this trial. We had around 22 mice that were fully cured of their tumor with one treatment of our compound. We waited for a couple of months, and then we decided to check whether these mice have built an immune memory against this particular tumor. So we re-challenged all of these mice with the same type of cancer cells. And as you can see here, every single mouse that got re-challenged rejected the tumor. Well, you can see that some of the mice clearly did take the tumor. It grew for a little bit, but the immune system kicked in and cleared it out fully. So what that tells us is that 22 out of 22 re-challenged mice fully regressed their tumors, and it's telling us that there is some sort of a memory that happens. But just to confirm it to ourselves, we decided to compare the rescue that we see in the immune proficient mouse, the BALB/c mouse, against an immunocompromised version of that mouse model. And for that, we relied on NUDS/KIT gamma chain mice, NSG mice. And there, when we implant the tumors, treat them the same way with PBS, the cancers take off very fast, and within a short period of time, they get to very high volumes. Then in these mice that are immunocompromised, when we treat them with a single dose, the high dose of our compound, we still manage to cure 1 out of 8 mice, but the majority of the mice, some of them experience some delay, but what we think is happening there is that that single treatment of our compound kills 50, 60, 90% of the cancer cells. In the one lucky mouse, basically, we killed 99% of most of the cells there, so they didn't have a way of coming back up. But then in the other treated mice, maybe we didn't kill all of the cancer cells at once in that single dose. And the remaining cells, basically, because they didn't activate an immune response, because there is no immune response in these mice, eventually came back and the tumor came back. So the one thing to note, obviously, is, there is a delay in the tumor volume, but eventually once it takes off, it takes off. And that's very consistent with us having been able to kill a lot of the cells first, but we didn't get engagement of the immune system. So to summarize all this, what we think our compound is doing is that it has a cell intrinsic activity on the cancer cells, cytotoxic cell intrinsic activity, by exploiting the high amounts of RAD51 inside the cancer cells. We're releasing that calcium out of RAD51 in the cancer cells, and that kills the majority of the cells. But those cells that do die, they don't just die, they actually die and present a lot of immunogenic markers on their surface, and that leads to the professional antigen-presenting cells to come up and gobble them up quickly and present them to the immune system, to the T cells, to train the immune system to actually have a memory against the neoantigens that are present in these cancer cells and have a prolonged response against those tumors and curative response against those tumors. So this is, in a nutshell, the program, the RAD51 program. It's triggering a novel immunogenic cell death that kills cells in a very quick way, but doesn't just kill cells, it also engages the immune system. Our medicinal chemistry efforts led us to having developing a compound that's very stable and a compound that allowed us to do a lot of in vivo work. And our activity, the activity of our compound can turn very cold, immune checkpoint cold tumors that are refractory to treatment with Opdivo, Keytruda, or any of these types of checkpoint blockade inhibitors therapies. We can sensitize these cells with a single treatment with our compound. And the ongoing studies right now are the second generation version of this compound that allows us to do systemic administration of the compound, and also deep understanding of the mechanism of action, the immunological mechanism of action of how we turn cold tumors into hot in response to treatment with STX100. Today I've had the time to talk about RAD51. I know I'm past time, I think. So I just want to share with you that we also have a oncogenic KRAS program, as I said in the beginning. We have a couple of other programs. We're very excited about the power and strength of our platforms and the new biology that they allow us to discover. And from here, I'll take any questions you have. Thank you.
So for those of you who are still with us, if you could keep sending questions in. If we can't get to them during our normal scheduled programming run here, we will certainly answer them afterwards, because Charlie and I will be available right after the talk in the excuse me, in the networking lounge, and we can continue to answer questions there. Charlie, assuming that you can hear me, could I ask you just a couple of questions?
Of course.
So I'm very, very taken by the fact that in your C26 colon model, and actually I should preface this by asking, so were these tumors generated from a single flank injection of tumor cells?
Yeah, they were, yes.
And so as far as treatment was concerned, were those intratumoral injections or dermal?
No, they were actually intratumoral injections. It's particularly because the peptides tend to have a very short half-life. We've managed to stabilize the peptide and make it very proteolytically stable.
Right.
But when we do systemic administration of the peptide, it clears very fast through renal clearance, basically. Even though the peptide remains intact, it clears through the kidneys. And to avoid having any issues of pharmacokinetic property weaknesses that prevent efficacy, we decided to go the intratumoral route just to probe the biology first and understand what happens when tumors inside an animal gets exposed directly to the treatment.
Right. So given that, then it's actually even more remarkable, seeing that the heterogeneity that you find in a tumor, regardless of whether I think you have a single, ostensibly single clone of cells going in to develop a tumor. The uptake then of the peptide, if clearing is fast, the uptake of the peptide must be remarkably fast, and the therapeutic effect of the peptide then even though you may not see a decrease in tumor volume occurring for a certain number of days. As I say, the uptake must be remarkably fast and to virtually all cells in the tumor.
Yeah, well, that's pretty much what was so impressive to us, that we are seeing inside the animal what we see in the dish. That
exactly.
the peptide is being able to enter to the cells within a couple of minutes. So obviously the scarring around the tumor, the cells die, but the scar is still there. So we don't necessarily see a resorption of the tumor within the first couple of days. But we do clearly see that it never grows back up. And yeah, I mean, I agree with you. I don't think that whatever leftover of the peptide that was injected, that didn't get taken up by the cancer cells in the first few minutes, in the first half hour, let's say, is not going. Just 1 second, I'm being asked for. Sorry about that. We had a repair person needing to enter. Yeah, so the amount of peptide that stays in the mouse, systemically in the mouse after the first half hour or an hour is very, very little. So whatever doesn't go in within the first half hour is not going to go. I mean, whatever cells did not take up the peptide within the first half hour are not going to take it because it's a single treatment. And yeah, so that to us, like what we saw with the not-skid gamma chain knockout mice, is that we do see a strong delay. And there's one mouse that completely got cured in the absence of an immune system. Presumably, as I said in the talk, because maybe in that particular tumor, every single cell, or the 90 or 95% of the cells in the tumor that was implanted, took up the peptide from that single injection and got killed by it. And then there was no need to engage an immune system to get rid of all of it. While the other tumors in the NSG mice, they experienced a very prolonged delay in tumor volume takeoff, but eventually they took off like after a week, which to us suggests that like we've killed the majority of the cells in that tumor when we got the intratumoral injection. But then there were a few escapers that didn't take up the peptide. It's not that they're resistant to the peptide; they just didn't take up the peptide before it cleared. And those obviously now there's no immune system and there's no peptide around, so they're going to take off and keep going. In the immune proficient mice, the same phenomenon is happening in like that window of time that when they take the peptide, a lot of the cells are going to be cytotoxically killed by the intrinsic activity, the cancer intrinsic activity of the peptide. But then what happens next is that dendritic cells and macrophages come in basically to clear those dying cells. Right. And in the process, they're presenting their proteins to the immune system and having basically building like vaccinating the mouse against that particular tumor. And what we see also, so obviously we didn't just rely on the CD26 colon cancer model. We've done mammary tumors, the 41 model, melanomas, B16F10 model. These are very difficult models. And one thing we consistently, so we see activity against all of them, but there's a very clear consistent correlation between the known number of neoantigens in the cancer line. Like for example, CD26s are known to have around 2,000 neoantigens relative to CD1 or biopsy mouse. The B16s have much less than that, like maybe 500, 400, 500, and 41s are single or double-digit type numbers, very small numbers. So the less neoantigens the tumor has, the weaker response we get. We do get responses from all of them, but it's harder to get the immune system engaging with the tumors that have less neoantigens.
You're right. Yeah. So I think we have. I do have another couple of questions, but let's. I'll hold those until we get to the networking room. And so as far as our time in the presentation is concerned right now, we'll wrap it up. And excuse me, thank you very much for those of you who have attended. And I hope you can join us in the networking room for at least another, say, 5 or 10 minutes, so we can chat some more and potentially answer additional questions. So thank you very much.